From the Library of Congress in Washington, D.C.
Welcome, everyone. I'm Tomoko Steen at Science, Technology, and Business Division here at the Library of Congress. Today's event is sponsored by our division. It's my great pleasure to introduce distinguished speaker today, Dr. Giorgio Trinchieri. Dr. Trinchieri is the director of the Cancer and Inflammation Program and the chief of the Laboratory of Experimental Immunology at the National Cancer Institute. NCI is a part of National Institute of Health, as many of you know, and research focusing on the cancer is conducted. Dr. Trinchieri received a medical degree from the University of Torino in Italy in 1973. He was a member of a Basel Institute for Immunology in Switzerland and an investigator at Swiss Institute for Experimental Cancer Research. You can see the detailed biography in the handouts right there if you're interested, and has also most recent publication on the reverse side. After moving to here in the United States, Dr. Trinchieri and his team discovered a molecule called interleukin-12 in 1989. Interleukin is a key component for immune system. Especially this interleukin-12 involves infection, managing infections, and also autoimmunity. So, for example, the MS, the multiple sclerosis, and irritable inflammatory bowel disease type of mechanisms also involved by the interleukin-12. One of the most recent research he has been doing is human and all other animals' gut system. In your gut system, you have microbiome, there's a variety of bacteria in there, and controlling your health. As an expert in immunology and also medical doctor, Dr. Trinchieri is today talking about your gut system, immunology, and also cancer. So before further ado, please join me welcoming Dr. Trinchieri.
Thank you, Tomoko. Thank you very much for inviting me here. Thank you all of you for coming. And what we're going to talk today is the effect of microbiota on health and disease. And I will try to introduce you what's the meaning of microbiota and what can be used, and this knowledge can be used in medicine, both for disease prevention and disease therapy. In the 17th century, the Dutch drape maker Anthony van Leeuwenhoek liked to play in making lens, and he built some of the first microscope that show here. And by this small microscope, it was probably given just a magnification of few fold. He was actually the first one to describe microbes. This is the way that he drew them. And he looked in the plaque between his own teeth, and the way it's described in the letter to the Royal Society in London is then I almost saw a great wonder that in said matter there are many little living animalcules very prettily moving. So that was really the first description of microbes and the first description that this microbe can live actually in our body. We made big progress, obviously, technology. We can look the microbes in our mouth as well as other sites of the organism. Microscopy has been advanced greatly. And also, through the year, we learn how to culture the microbes, the bacteria in vitro, in Petri dish. But even if this technology is improving very much, we can still only culture a fraction of the bacterial species living in our body. So the real progress in understanding this population of microbes in our body and in general is been with the progress in DNA sequence technology in the last 10 years or so. Obviously, that technology was very much involved in the identification of the human genome and the human genes. And with this technology, now we can really characterize the gene in a population of microbes and understand the whole component, regardless of the fact that we can isolate and grow them in vitro. So now we define as a human microbiota the ensemble of microbes that reside in our body. And the human microbiome is the ensemble of genes that are present in this population of microbes in the microbiota. So obviously there's been incredible progress, scientific progress from the 17th century today. Obviously the good news that this new technology, molecular technology, system biology, now we can really start to understand what Van Leeuwenhoek saw under this primitive microscope. The bad news that this new technology generates an incredible number of information that become very difficult to interpret, to analyze. We need really computer tool, bioinformatic tool to be able to understand, and the task to identify this is really an enormous task. Still, we have made major progress in the understanding the role of the microbiota, both in health and disease. But if we really want a full understanding of the mechanisms, the molecules that are involved, and use that, apply that for prevention and treatment of disease, we really need to continue with larger study, and particularly should be coordinated multidisciplinary effort, because we need now different scientific disciplines to get together and allow us to understand this complexity. So I want just to give you an evolutionary idea of what's happening is obviously the microbes and bacteria were the first organisms present for billions of years on the earth. And in more than 2 billion years ago, something happened, and bacteria start to join each other, what is called an endosymbiont, and form organisms from two bacteria. And the cell type, the eukaryotic cell type, is the one that form all animals, including ourselves. They really derive from the fusion of a type of bacteria-like cell, it was called archaea, that phagocytosed engulfed another bacterium that was able to fix oxygen, and that became the mitochondria in eukaryotic cell. This allow our cell to utilize oxygen to breathe. And eventually, these eukaryotic cells start to form multicellular organisms, the early metazoa. And these organisms, primitive organisms, like still today, we live in a sea of bacteria. Bacteria and microbes are everywhere. So they form a symbiont with the multicellular animal, with the bacteria. And some bacteria we define as symbiotic or commensal because they don't damage, but actually they help each other. They live together and they help each other. But some, as we know, some microbes, some bacteria become pathogenic. They can cause disease, and the organs need to defend against these disease-inducing microbes. And other that have been defined more recent are called pathobiont. Now, pathobiont are microbes, the bacteria, they're present normally in our body, but because of the equilibrium in the population of bacteria we have in our body, they don't create any problem. But this equilibrium is broken, and these bacteria can expand, they become in larger number, change some of their genetic characteristics. They can become pathogenic, they can induce disease. Now, the interaction between the host and the commensal, this crosstalk, especially in the early organism, was very important for the metabolic regulation in the organism, for morphogenesis, for the formation of the different organs, the different parts of the organism, regulate physiology in general, homeostasis, and they're also involved, because of the interaction between microbes and the organism, in the mechanism of resistance to infection with the pathogen. Those bacteria become able to induce disease. And very early in evolution, there was a specialized cell type that became very important in this type of interaction with the commensal microbes and the resistance to infection. And these are the phagocytes that were described by Mechnikov in the 19th century in Daphne and starfish larvae as a mobile phagocyte. These are basically cells that are able to phagocytosis. They can engulf, they can eat, for example, bacteria and destroy them. So they really play a major role in this crosstalk between the microbes and the organism. And in our body now we know, in medicine we now know this very cell very well. They are called monocyte, macrophage, and neutrophils, and other cell type. They play a major role in immunity to infection. Now, when we move to this very simple early metazoa to the higher vertebrate in human, there's this crosstalk between the commensal and the host that remain very important, is play a major role in the resistant immunity to infection, but still have a role in regulation of our own metabolism, regulation of physiology, homeostasis, and a minimal part, but still some role in formation of our organ in morphogenesis. So the basically bacteria and other microbes live on all the barrier surface in our organism. Barrier surface are those like the skin, or the lung, or the intestine. Those are the surface in which our organism communicate with the outside environment. And in all these surfaces, there are microbes, there are bacteria, but they're all quite different in the different type of the body. So the mouth, pharynx, and the lung, the respiratory system, they are different bacteria. They are present on the skin or present in the digestive system, in the gastrointestinal system. And in gastrointestinal system, particularly in the lower small intestine and in the colon, is where the larger mass and incredible mass of the microbes and bacteria in particular, and they play a major role in this interaction with our interaction with this commensal bacteria. So we co-evolve with these microbial partners. We really have a composite of species. We have our own human cell, obviously, but we also made of bacteria, archaea, fungi, virus, bacteriophage. As I mentioned, they inhabit all the barrier surface of our organism. And they actually outnumber the human cells present in the organism by about 3-10-fold. And if we look at the gene, their DNA, their microbiome, that's contain approximately 100 times more gene in our own human genome. So the microbiome is really integral part, not only of our body, because they live in our body, but our genetic landscape and our general regulation of the our physiological equilibrium, these host homeostasis. So if we look ourselves in the mirror, and we could have the von Leeuwenhoek microscope or something, we could see that our skin is really covered with bacteria. It's not something we should worry about. It's not something we should try to wash out. They're actually absolutely needed. We cannot live without our own bacteria. And we should really look ourselves as a meta-organism that's composed by our cells and this microbe. And with their own gene, their process, their metabolism, and they work in concert. In the meta-organism, both our cells and the microbial cell act as a sensor for change in the environment. That could be changing the temperature, changing in the type of food we eat, infectious agent. And they exchange, they talk each other, they exchange information, and they adapt. They have a response, they are adaptive or immune response to the change in the environment. And what is important to consider that if environment change, our own gene cannot change. Our genes are fixed. I mean, they are very constant. But the microbiome can change because the microbiome is made by hundreds or thousands of different species of bacteria, and when the environment changes, they can change their number, their proportion, and therefore their genetic change completely, and they can adapt and respond to the change in the environment. Now, in this commensal crosstalk in the last few years has been clearly shown in the metaorganisms that regulate many of the functions of the organism. I mentioned to you regulate metabolism, regulate cardiovascular and muscular function. It's even been shown to have a role in regulating neurological behavior and cognitive function. It's definitely play a role in aging, in the formation of blood cells, or hemopoiesis, regulate what is called the circadian rhythm, the change in the organism, physiology of the organism through the day, and particularly regulate inflammation and immunity. And through this role of inflammation and immunity is clearly an effect on cancer initiation, cancer progression, and response to therapy. The NIH recognized about 6-7 years ago the important role in microbiome. They started a major project that was really to develop new technology and the catalog of basically the bacteria present in our body. So this project that was they funded, I think, approximately 10 centers, 1 intramural and 9 across the nation. And they characterize different sites of the body, the type of microbe they are present on the body. Where we get the microbe? When we are born, the fetus is actually sterile. They are not microbe in the fetus, but then at delivery, the newborn acquire bacteria. And the type of delivery affects the type of bacteria we get. So the vaginal delivery or the C-section, because the newborn is exposed to different anatomical sites in the mother, get colonized with different bacteria. So there are different starting microbiota depend on delivery. But then during early childhood, there is acquisition of many different species from different sources, from the environment, from other people, from the family, and there is a rapid change and diversity in the microbiota. But eventually, when you get in the adult age, that gets sort of fixed and is maintained fixed until old age. But can change. The alimentation can change it. Disease can change it, obesity can change it. So there can be some variation, but it tends to be rather constant through life. It definitely changes in old age and probably is associated with the aging process. So what can change our microbiota? Our own genetic, our own gene is clearly affecting that. I mentioned the mode of delivery affect the composition of microbiota and the age affect the microbiota. There is also major difference due to geographic variation. And that can be for different reasons, but the geographic location usually affect the lifestyle, the nutrition, and the hygiene, and that is going to affect the composition of the microbiota. Also, we can take food supplement like probiotics. We can treat with antibiotics. We have disease, especially infection. Then they can affect temporarily and often permanently the composition of the microbiota. And that can bring to what is now defined as dysbiosis. That is an imbalance in the microbiota that changes what is considered a normal, healthy microbiota. And that may often to be associated with this different type of sickness, of illnesses. So what the gut microbiota is doing, I told you metabolic function. For example, they are involved in the synthesis of vitamins, the metabolism of bile acid and host hormones, fermentation of food, particularly of carbohydrate. But I mentioned before, they're also important in resistance to infection, and they can prevent, by competing other organisms, they can prevent colonization by pathogens, or the overgrowth of the phytobiome, this normal microbe that the overgrowth can cause disease. They contribute to fortify the barrier, the cells that divide, the cells that line the gut and divide the lumen, the gut where bacteria are, from the rest of the organism that is supposed to be sterile. And they also signal to our own immune system, and they set a tone the immune system for the immunity that give the resistance to the infection. So if we look at the gut microbiota, I mentioned there's an effect on immunity that can be local in the mucosal immunity. It can affect the pathology at level of the mucosa, for example, the inflammatory bowel disease. Can have an effect of distance. I mentioned metabolism. The gut microbiota has a major role in the regulation of body mass, can because of obesity, metabolic syndrome, inflammatory metabolic syndrome, insulin-resistant type 2 diabetes are very much connected to change in the microbiota. They also regulate systemically the immune system, development of the immune system. And for example, that's seen by the fact that the gut microbiota is needed for in our lung to resist to flu virus infection. Without gut microbiota, our ability in the lung to resist the infection like flu is very much decreased. Also, autoimmune disease, the disease in which the immune system attack our own organism and cause disease, is needed the gut microbiota, for example, in autoimmune disease in the brain, in the joint, or in the liver. What change when we don't pay attention to the change in the microbiota? If we antibiotic use at early age, there's been shown to induce dysbiosis, and that may predispose to obesity. It's been clearly shown in experimental animals like mice, but there are also epidemiological data show that infant less than 6 months get treated with antibiotic predispose them to an increased body mass by the time they get toddler, and with the increase of their weight and obesity. The other correlation is with allergic and autoimmune disease. And for example, there are kids has been given a certain number of antibiotic treatment have a significant increase in the risk of chronic colitis, inflammatory bowel disease. And if kid has been treated with more than 5 courses of antibiotic, that risk can be up to 3-fold increase. There's a major increase in this type of disease. And indeed, the inflammatory disease that affect the barrier tissue are on the rise. There is now in the state about 1.4 individuals suffer for a chronic colitis. 9% of the kids develop food allergy by age 5, and asthma affect 1 in 12 individual. And the so-called hygiene hypothesis, the thing that we tend to eliminate exposure to bacteria or to use antibiotics and so on, is considered one of the hypotheses. This change increase in this disease that observed in the US, in civilized country, and less in underdeveloped country or more historical in humanity. So we really need to be careful the way we change the microbiota. And obviously, antibiotics are needed. I mean, we want to stop infection disease, but they often, especially a broad spectrum antibiotic, they often kill beneficial bacteria together with the pathogen, and one really have to pay attention to try to avoid that. So I told you we're going to talk about cancer. So cancer grow in our body, and we are a meta-organism. So you can see that cancer is not only a disease of the body, it's actually a disease of the all human meta-organism. And as you all know, that the cancer cells are genetically modified. Genes in the cancer cell are altered. There's oncogene, suppressor gene, and suppressor tumor gene. And this change in the genetic change in the cancer cell is what makes the cancer cell grow faster and not being subject to certain the ability to escape from growth control mechanism. However, the tumor cells will never grow in organism, will never become a tumor and kill the organism unless they found the right environment, the right tumor microenvironment. And in this microenvironment, the inflammation and immunity play a major role in creating either a pro-tumor or anti-tumor environment. And all of this, as I mentioned before, inflammation, immunity, and the composition of this tumor microenvironment, the factors that are produced there are regulated, may be modulated, regulated by the microbiota, particularly by the abundant gut microbiota. So to the different effect of gut microbiota mentioned before, we need now to add the effect of the gut microbiota on the tumor growth. We know there are bacteria that have been shown to be carcinogenic. The only one is actually officially accepted as carcinogenic by WHO, is Helicobacter pylori for stomach cancer. But other, like Escherichia coli, Fusobacterium, has been associated and proposed to be important in colorectal carcinoma. Infection, especially in the East Country, with Salmonella enterica, seems to be associated epidemiological with gallbladder carcinoma. And there are a number, 7 or more oncogenic virus or even parasite, worm and so on, that can be associated with cancer. And in general, about 16 or more % of worldwide cancer are now known to be caused directly or indirectly by infectious agents, infectious microbes. In addition to that, the gut is the ability to regulate the growth of tumor that are not infected, that do not contain bacteria. And for example, there are data, at least in experimental animal, for mammary breast carcinoma, for lymphoma, for sarcoma, for ovarian cancer, in which the commensal microbiota can change the growth of the tumor. And in general, one can say that it's likely there's modulation by the gut microbiota of cancer progression, as well as on the response to immunotherapy and chemotherapy. Now, all of that is because of the role of, it's been studied very much in the last 10 or 20 years, the role of inflammation and immunity in regulating the growth of cancer. And inflammation immunity really is involved in a predisposing condition, like obesity, for example, is involved in this in facilitating those genetic alteration that we see in the basis of the cancer cell transformation. But they're also involved in promoting the growth of the cancer, this ability to form metastasis, the type of immune response against the cancer that may be induced, and also I will describe you some of our study that involve in regulating the way that the organisms respond to cancer therapy and the way the therapy can be effective in eradicating cancer. And all of that, as I mentioned before, can modulate in different way by the commensal microbiota. So the way that we did to study the effect of the intestinal microbiota of cancer was to induce a growth of subcutaneous tumor in mice. And then we use 2 types of cancer treatment. We use immunotherapy with a substance that's called a CpG oligonucleotide, or chemotherapy using some of the chemotherapeutic agents that are commonly used in the clinics, like the platinum compound oxyplatinum, cisplatinum. We treat the tumor of conventional raised mice, or with mice in which we use a broad-spectrum oral antibiotic to eliminate mostly completely eliminate the gut microbiota, the gut bacteria. An alternative, we use germ-free mice. Germ-free mice is mice that are kept from their birth in a completely sterile environment, so they are not associated. They have no, they are not bacteria, no germ that live in them. This is the type of response. What we are showing here is the survival of mice with tumors. If we don't treat them in this type of tumor, show you on the left for the immunotherapy, they die in about 2 or 3 weeks, all of them. We treat them with this compound called CpG, and we can block the tumor and save up to 70-80% of the mice. So this treatment is very efficient in the mice. But if we treat with CpG together with antibiotics, when we can protect only about 20% of the mice. And the result with chemotherapy, I'll show you oxyplatin, is very similar. In this tumor, mice die in about 10 days or less, but 80% of them survive when treated with oxyplatinum. But if we treat with oxyplatin and antibiotics, then only about 20% of them survive. Like to try to explain you a little bit of the mechanism, try to be very as simple as I can. This is a subcutaneous tumor under the skin that in mice that we not treated. We treated them with CpG. In a few hours or couple of days, you get this necrotic tumor. The tumor basically is all dead. That's basically almost completely just a scar. But if you do the same experiment in mice that get CpG and antibiotics, you see the treatment is ineffective. You don't see this scar, this necrosis. This is due in part to the production of the substance in the body of the treated animal that's called a tumor necrosis factor, because it can induce necrosis of a tumor. If we use mice that are genetically not able, they don't have the gene for this tumor necrosis factor, you see these mice are resistant, they do not respond to the treatment. So the mechanism that you should think that when you have a tumor, as I mentioned before, is not only the tumor cells, but they are infiltrating inflammatory cells and other cells, including this famous phagocyte, macrophage, neutrophils that I described before, that really the major cell type that is interaction between the host and the microbes. So when you inject CpG in the tumor, what CpG is inducing activated is a phagocytic cell and induce inflammation in the tumor. And one of the factors that's made by this phagocytic cell is the tumor necrosis factor that kills the tumor cells. It is also induce an immune response against the tumor, and in most animals can completely eliminate the tumor. If we treat with antibiotic, we block the ability of the animal to make TNF, block the ability of the animal to make this immune response, and eventually the tumor will grow and control. Even the treated animal will not be able to control the growth of the tumor. We'll show you now data that we obtain when we completely eliminate the gut microbiota, but we want to know which bacteria, which species are able to affect the response to the tumor. So we sequence the gut microbiota, and we correlate the presence of different species with gut bacteria, and we found some that are associated with an increased response to cancer therapy, and some that were associated with a decreased response. Two of them, we grow them in vitro in petri dish, these two species called Alistipes shai and Lactobacillus fermentum, and we then gave them orally to the mice before we treat them with CpG to see the effect on the tumor. And we found that one, the Alistipes, was actually, when we increase the response of the therapy, so the tumor is destroyed better. And another one, the Lactobacillus fermentum, is actually decrease the ability of the CpG to kill the tumor. So really showing that not only the microbiome is needed for the response, but we can modulate that and try to modulate the type of therapy response that we get. The oxyplatinum mechanism is a little bit more complicated. Is that based on the fact that antibiotic-treated mice do not produce chemicals that are known as the reactive oxygen species when treated with the mice are treated with oxyplatinum. So we have again the phagocytic cell that I've been talking till now. They produce, when they are activated, they produce reactive oxygen species like superoxide or hydrogen peroxide, or hydroxyl radical. And that's a very important function in the phagocytic cell, because that's one of the mechanisms the phagocytic cell used to kill the bacteria they engulf. But there's these hydroxyl radical and ROS are also toxic, and they can actually induce DNA damage and damage cells. Now, in the case of the oxyplatinum, what oxyplatinum is doing is binding platinum to the DNA of the tumor cells, and in this way alter the DNA conformation. But it's also induced the formation of ROS, activated phagocytic cell to form ROS, and in order for oxyplatinum to kill the target cell, that is done by affecting, by damage the DNA of the tumor cells, both this platinum bind to the DNA and the ROS production is needed. So what happened when you treat the mice with antibiotics? So we've been measuring, the experiment I'll show you on the right, measure the production of this reactive oxygen species by the ability of inducing a substance we give to the mice that emit light, become luminescence when ROS is produced. So if we give oxyplatinum to the tumor-bearing animal, you can see that we get this light where the tumor is, showing the ROS is produced, reactive oxygen species are produced in the tumor. But when the mice has been treated at the same time with antibiotics to reduce the microbiota, then there's no production of ROS. So if that is blocked, this ROS production of ROS, even platinum still bind to the DNA, there is no DNA damage, there's no tumor toxicity, and the chemotherapy is not efficient any longer. So what I've been showing to you is that the gut microbiota and other microbiota, but particularly the gut, affect this phagocytic cell, the myeloid cell that are infiltrating the tumor, and CpG through the production of this factor, that is a tumor necrosis factor, oxyplatinum regulating the production of ROS. But it's now known there are many other tumor therapy that are affected by the microbiota with similar mechanisms involving the phagocyte. And these include two type of tumor therapy has been very much in the news recently, the adoptive T-cell transfer and the immunocheckpoint inhibitor therapy. And the reason it's been in the news in the last 2 or 3 years, because these two type of therapy are the only, in history for the first time, treatment has been shown, at least in a significant proportion of patients, to be able to really induce complete tumor regression and long-term survival in patients, for example, in melanoma patient, in renal cell carcinoma patient, and in bladder carcinoma patient. And that was the reason why Science Magazine last year recognized cancer immunotherapy as the breakthrough of the year, the scientific breakthrough of the year. And of interest, one, the runner-up breakthrough is actually the effect on the microbe on the health, including the effect on the microbe on the cancer therapy that was published in 2013. And I would say that a major contribution to this success, of scientific clinical success, is provided by NCI intramural research program, as well as largely by NCI-funded laboratories across the nation. So NCI had really a major role in supporting directly or indirectly this scientific and medicine progress. So how can we change the microbiota? How can we change or reverse this dysbiosis? Well, we can use antibiotics. Antibiotics are a problem because they can induce dysbiosis, but if we are careful the way we use them, we use more selective antibiotics, we may affect and change and try to reconstitute a healthy microbiota. We can use probiotics. They are live microorganisms that may not be constituent of the host microbiota, but they may change the equilibrium microbiota and may confer a health benefit. And the other way is use what are called prebiotics. These are basically non-digestible food components like fiber they find in many different type of food, and they favor the growth or beneficial member of the gut microbial community and try to fix this dysbiosis. But the other thing that received much interest in the last few years is the fecal microbiota transplant. Now, this is a very empirical type of medical treatment. When basically, when there is dysbiosis, a disease associated with change in the microbiota, one can try to give the microbiota from a healthy individual, and this is basically fecal microbiota, get clean and filtered, and then is delivered either by colonoscopy, endoscopy, in the lower intestine and in the colon of the patient. So this is based obviously in the idea that if the person is healthy, his microbiota is healthy. But unless we know exactly what's healthy microbiota, that's a very empirical approach. Still received much of interest. It was used in some type of infection. I will describe better the Clostridium difficile, as well as attempt to use for chronic colitis, for inflammatory bowel disease, Crohn's disease. Obviously, it's an empirical method. There's some risk. Sometimes could induce more inflammation instead of less inflammation. It's actually regulated as a drug by FDA. And but I want to talk about Clostridium difficile because this is really a proof of concept, a clinical proof of concept, how this approach to change the microbiota may be important. Clostridium difficile is a bacterium that causes, this is really a pathobiome, that cause syndrome range from diarrhea to life-threatening inflammation of the colon. And illness from C. difficile infection affect usually older adult in hospital or in community, and typically after use antibiotic medication or protein inhibitor for reducing stomach acidity. And it's quite serious disease. As there are about 3 million new cases in the United States every year. It's a chronic disease. It's difficult to eradicate and cause about 30,000 deaths every year. Now, the fecal transplant, even this very empirical process actually cure, and within 1 or 2 days, more than 95% of the patients. And if we look, what I show you on this slide, that each bacterium species present in this tool is indicated by different color. You don't need to know what it is, but this is a patient, and this is the healthy donor for the fecal transplant. You can say that their composition are quite different. And if we look after the transplant, 2 weeks or a month after the transplant, you see that it's really a permanent change. The microbiota, the fecal microbiota in the patient is much more similar to the one of the donor than the original microbiota of the patient. As I say, this really within 1 2 days completely blocked the response, the infection with the C. difficile. Now, this is very empirical. We really don't want to do that for a regular treatment in large number of people. We might not know if we are really using a healthy microbiota or not. But important study are the one that really start to attempt to try to identify, like I showed before for cancer, which species are involved in this regulation and blocking the infection with Clostridium difficile. So what I'm showing here is a recent work by Eric Palmer at Sloan Kettering in New York. And he found out that in the normal microbiota, there is an equilibrium between different species, competition between different species. And another Clostridium, the Clostridium scindens, is actually metabolizing bile acid in the intestine of the patient, of the individual, and this blocks the growth of Clostridium difficile. If you use broad-spectrum antibiotic, you reduce the diversity of the microbiota, you eliminate the Clostridium scindens, and then the Clostridium difficile can grow and cause the disease and even death. And Dr. Palmer has actually been able to show, at least at the moment in mice, that he can very efficiently, by just giving one bug, the Clostridium scindens can cure most of mice, and even better if this bug together with 3 or 4 other bacterial species, so trying to give an equilibrate mixture of bacteria, but still by itself, the Clostridium scindens is enough to block the infection with Clostridium difficile. Now, Clostridium difficile is really an example of antibiotic resistant or partially resistant drug. What are they called? The superbug that represent a major problem especially in the hospital setting in antibiotic-treated patients. And some of them, they are very common in hospital setting. Some of them you know very well, very famous, for example, the MRCA, the methicillin-resistant Staphylococcus aureus. And we have, 4 years ago, a very typical case of that in actually an IH clinical center. In 2011, 12 patients died of hospital-acquired infection with the antibiotic-resistant Klebsiella pneumonia. And that was studied. I show you with the genomic sequence method that really now we can study the microbiota. And the NIH team that is a different institute that have studied this infection in NIH and stopped it and identified the source and stopped the infection was Segres, Nikin, Palmore, and Anderson, was recognized with the American medal, Service to American medal, as the first ever use of this precision medicine, genomic medicine, to stop an hospital-acquired infection. And we are obviously trying to do in a National Cancer Institute a similar type of approach. We established a new facility for both sequencing and bioinformatics study, and we want to screen the microbiota of cancer patients that undergo either chemotherapy or immunotherapy, identify strain that would favor decrease anti-tumor response, and see if that we can either predict the type of response we'll get to therapy, or even try to change the microbiota and make the response to therapy, particularly to immune therapy, the most efficient one. So if you think about an infection disease or cancer growing in an organism, it's really not different from an invasive plant that's growing the wood and is eventually suffocating and destroy the wood. So how we cure that? Well, the typical medicine, especially in oncology, is being the war on cancer. That is, medicine is a battlefield strategy. The human body is a battleground. This is very efficient. You kill the invasive, but you also kill the wood when you do that. The other is medicine is park management. We should have a target removal of invasive. We need to eliminate the pathogen. We need to eliminate the cancer cells, but really pay attention to restoration, promotion of native species. And what it is in the patient, the cancer patient infection, is really a thing what is immunotherapy and maybe the microbiota study are pointing to. We need to make sure when we cure the patient, we eliminate the tumor or eliminate infection, we keep a healthy immune system, we limit inflammation, and we try to correct the composition of microbiota in general, the physiology of homeostasis of the organism, and they're really a healthy organism and not an organism to be debilitated by the type of therapy. And this is really what has been just part of it has been announced recently by President Obama and by the NIH is the new effort in precision medicine. Precision medicine is really based a lot on these new technologies available. The SEML technology has been showing you, the genome sequencing, the really careful characterization of the patient, the immune system of the patient, of the genetic, both the microbiota individual, the genetic of cancer, and be a real very tailored and personalized medicine to try to get the most effective response for each single patient. So I would like to thank the collaborator in this work. My lab that done some of the cancer work has been showing. Amiram Duchev is actually in the audience. I've not been doing work yet with Colm, sitting over there, but Colm is actually the one that is setting up this new facility, and we work very hard on that. I'm important collaborator, Yasmin Belkaid at the Allergy Infection Disease Institute, Karen Frank in the Clinical Center Microbiology Service, and my clinical collaborator in the NIH Clinical Center, Heidi Kong and Jim Galley. Thank you very much.
I hope I can present my question sensibly. With respect to Clostridium difficile, I had read a few times that its growth is accelerated when various stomach acid reducing medications are given to the patient. And I'd like your comments on that.
Yeah, this, usually is such that I said the question is whether the Clostridium difficile infection is favored or induced by the use of antacid and in the patient. And as I said, the major cause is usually been attributed to the use of antibiotics, but the antacid and particularly the proton pump inhibitor, they clearly change the utilization of the food, and they can change the microbiota. And it's been clearly shown they change the microbiota, and they can affect the growth of the bacterium. Also, as was shown by Eric Palmer's study with this competition between two species of Clostridium, there's actually what Clostridium is protecting is actually digesting bile acid that obviously come from the stomach and digesting to a form of processed bile acid in which the C. difficile bacterium cannot grow. Now, if you get an increased amount of the change in the bile acid and change in this process of this microenvironment, then the Clostridium difficile can grow. So that is absolutely correct. The antacid, especially the proton pump inhibitor, the strongest antacid, can induce that.
So if one wanted to reduce or eradicate using the proton pump inhibitors, what would be a less drastic approach?
Well, obviously some of the traditional antiacids just buffer the acidity, or some of the less strong Tagamet and so on. I mean, they clearly they've been less associated with that. They are also much less efficient in reducing stomach acidity. So that's always the catch-22 situation. When sometimes we present the data on cancer, newspaper, and sometimes people were calling up and say, "Oh, we should not give antibiotic to the cancer patient." Well, cancer patient, especially during chemotherapy, they are infected with a very dangerous pathogen. You need to protect the life of the patient. You cannot worry about not giving antibiotics. So it's always this catch-22. Obviously, stomach acidity and ulcer and so on are again a disease of civilization. It's linked to stress and food and so on. So, I mean, this is very theoretical, but the way would be to change the predisposing condition and try not to treat. It's always better to prevent a disease than treat it. When you treat it, you have always this bad and good and bad side of the treatment.
Excuse me. Even though the room is not filled, I do want to thank you on your research into the microbiome. It's been a long time. And it's been an area that has not been focused on quite a bit, and I think there's a lot to really appreciate in what you presented here today and what will be coming in the future. My question has to do with what is the differences and the effects, and I'm less on cancer and more on autoimmune disease, on probiotics and how there are differences in the probiotics in changing the makeup of your microbiota. And the second half of that question is, what about the biologic medicines and their treatment, and have there been any relationships to their effectiveness, and or are there any side effects to using the biologics to treat autoimmune disease?
Okay, the question is what is the role of the microbiota in autoimmune disease, how different probiotic may be more or less effective, and what is the effect on the biological therapy on and affecting the microbiota and affecting autoimmune disease. The probiotic is very not regulated field, so it is really difficult to understand. Even when we buy probiotic on the store shelf, and there are so many billions of this bacterium, you really don't know how careful that's analyzed. You don't know if the bacteria are alive or not alive. There's no clear how long they stay on the shelf and so on. And not being FDA regulated, that there's really a difficult to say, get this one and not get this one that's available commercially, and I'm not competent to do that anyway. But there are components. Some of the probiotics being used that are known to have an anti-inflammatory effect, and I showed the data with the Lactobacillus fermentum in cancer, but what we get in that case is the Lactobacillus fermentum block the response to CpG because it decreases inflammation. So this type of bacteria is being shown in experimental animal to be able to decrease systemic inflammation and have an effect in some condition of autoimmunity. But I think this is a field that really need to be understood much better in order to really know what to take and what's working, not working. The biologic therapy is always something that's very close to my heart. That Tomoki has been talking about IL-12 and anti-TNF that I mentioned, or anti-IL-12, are now major drug for autoimmunity of different type. They are atomic bomb. They are really blocking substance that have a major role in our immune system, our physiology, and all these biologic anti-TNF as well as anti-12, for example, cannot be used in patients that may be a risk of tuberculosis, microbacterial tuberculosis infection, because they will reactivate infection. And it's because they change the way the immune system works. If they are used continuously, they are very likely to change the microbiota, and again, it may affect in a good or bad way what's the autoimmunity and what's disease, or induce different type of disease. And as far as I know, there's not many study about that, but I think would be something very important to study.
Could you talk about leaky gut and how that occurs and how that plays into this whole health and disease?
Well, as I mentioned before, one of the function of the question was discussed about leaky gut, the permeability of the barrier and how this may affect the disease. So as I mentioned before, what healthy microbiota is doing is really protecting the integrity of the barrier, definitely in the gut. We always consider that we keep our bacteria outside, but they actually can't go through the barrier, and they do all the time. You don't want that to happen too much. An healthy microbiota protect the barrier directly. The barrier is made, the epithelium, the cells that cover the gut is really a single layer of epithelial cell. And they have what they are called tight junction. They are very much attached one to the other, so not much can go through. Even there are mechanisms in which this permeability can be open and certain things can go through. The other thing that is comments that the microbiota is doing is inducing production of a substance I call antimicrobial peptide that and regulate the immune system, the mucosa, production of antibody, for example, IgA antibody, and that all work together in controlling the composition of microbiota and keeping the barrier intact. As when we start to have any pathology, an inflammatory pathology, or dysbiosis, for example, in chronic colitis or in infection like C. difficile, then this barrier is disrupted, and that obviously has physiological effect like the diarrhea and so on, but it's also increase the permeability of bacteria can come in contact with our organisms, can actually migrate to our lymph node, and that's really dramatically changed all the inflammatory environment. So that a change in barrier permeability in the digestive tract is not only affect locally, as I say, for example, symptoms like diarrhea, but is also having systemic effect. So or our health can be affected by that.
A couple of months ago, I have low vitamin D and high cholesterol because I had a blood test at my doctor for an annual physical. How can the normal person tell how your microbiota, how healthy you are at a particular point in time before you have a problem?
Oh, the question is, how do we know what is an healthy microbiota? Now we can say you have a problem, you don't have an healthy microbiota. Well, actually, that's a problem. A clinical problem with the microbiota is a clinical problem with the immunity in general. We always, when I talk about analyze the immune state of an organism, I always show a slide and say, if you have a heart disease, there are under very sophisticated tests that you can do and find out what you have. If you have, they want to know how your immune system is working, well, you get a blood sample and you count how many lymphocytes and monocytes are in there, and that's basically what is done. And you really, by doing that, well, it's a major problem, but you really don't know how good your immune system is. And we are in the same way with the microbiota because there was one problem of fecal transplant. They try with chronic colitis, with IBD. They say this person is healthy, so his microbiota should be good. And when they give with the patient, they actually they get more disease, they get more inflammation because that microbiota look healthy in the donor because this mucosa was okay, but in the patient is already inflammation that add to the inflammation. So that's exactly what we need to understand better, improve. Right now, you cannot really say what is good and what is bad. The mouse it's been helping here. It's not solving this problem, but it's helping because even mouse and human has different microbiota. You can actually take the fecal microbiota, for example, from fit and obese people, or obese twin even, so with the same genetic. And you can transfer that to germ-free mice, or mice have no microbiota, and basically make these mice have human microbiota. And the microbiota from slim person, you would get slim mice, simplifying the sperm. And the one from obese people, you get fat mice. There are some correlations, some changes, some species that we start to understand a little bit how they're correlated with that, but the full understanding of that will still require a lot of work.
This is a very good example of translational medicine, and please join me to thank Dr. Trinchieri.
Thank you.
This has been a presentation of the Library of Congress. Visit us at loc.gov.